Okay, what do you think, Angie? Should we go ahead and get started?
Yeah, I think we can get started.
Okay. Hi, everyone. Thanks so much for joining us today. I'm Courtney Scarborough. I am the Deputy Director here at NCES, and I'm really excited to have our speaker here today and excited to chat with you all. I'll just give a little introduction about what NCES is, and then I'll pass it off to Angie. NCES is the National Center for Ecological Analysis and Synthesis, and we're a research center that's affiliated with UCSB. And what we do is we bring people and data together to solve big environmental challenges. So rather than working in an isolated lab or in small groups, we bring together researchers, data experts, policymakers, communities to combine all the knowledge we have collectively and tackle questions in the environment that really no single person can tackle on their own. So we are currently in our 31st year. We've been around for over 30 years. And over that time, we've welcomed over 11,000 scientists and policymakers and individuals from around the world to come here and meet in Santa Barbara and really dig into these challenging environmental and social ecological issues. And those can be things from biodiversity to wildfire resilience to climate change to oceans. And we put together data and cyber infrastructure and create training mechanisms for people to learn how to solve these questions collaboratively and together. Um, so a lot of inform policy and management and other science as things develop. So this seminar series, we are in our sixth year of this seminar series, and we have Cesar here today as our second speaker this year. And this is one of the ways we like to share to connect science, to connect people with the broader community of Santa Barbara and beyond to bring this sort of information to everyone. So thank you again for being here, and please join me in welcoming our speaker and looking forward to having everyone here with us. Thanks so much.
Yeah, thank you, Courtney. And just a heads up, we have one more pres webinar for a part of the seminar series coming up next month in June. So we can share links for registration for that as well. But I'll go ahead and get started. And we can also share these stay connected slides a little bit later, so you can stay tuned with information and news from NCs. So with that, I'll go ahead and get us started. Thank you for joining us at our sixth annual NC Spring Seminar Series. Today we have the pleasure of welcoming Cesar Estien as our second speaker, as Courtney mentioned. And this year our series is centered around the theme of environmental data science and its impacts on communities and ecosystems. I'm Angie Garcia, a community engagement and outreach coordinator at NCs, and I'm joined by the rest of the seminar series planning committee. At NCs, we work to accelerate scientific discoveries that enhance our understanding of the world and benefit people and nature, as well as transform the scientific culture to be more open, efficient, and collaborative. Before we begin, we ask you to join us in acknowledging the Chumash people who are environmental stewards of the land on which NCs is located. So we pay our respects to the Chumash peoples and their elders, past, present, and future, who call this place their home. We acknowledge that this land on which we live and work are the traditional homelands of Indigenous peoples, including the Chumash peoples. This land today is the result of the erasures and exclusions of Indigenous peoples. As we come together to bring awareness to the issues of equity and justice, we recognize that the Chumash people and so many other Indigenous people and are fighting are fighting to dismantle the legacies of settler colonialism in their tribal territories. And as we continue to work towards diversity, equity, inclusion, and justice in data science, we strive to prioritize Indigenous data sovereignty and self-determination. Um, with that, just a little bit logistical information before we get started. Please feel free to add any questions that you might have during the talk through the Q&A button located at the bottom of your screen. And once the presentation is over, we'll have a moderated Q&A section, including these questions that are uh, added on by the audience. And I'll mention we also have some folks joining us in our lounge, so we invite to ask questions at the end. And without further ado, I'd like to introduce our speaker, Dr. Cesar Estien. They are an urban ecologist currently at Second Nature Ecology and Design, where they consult on how to merge nature and biodiversity with urban planning, management, and design. At Second Nature, Cesar works with nonprofits, private companies, government agencies, to envision greener cities that work for people and wildlife. Cesar earned their PhD in 2024 from the Department of Environmental Science, Policy, and Management at UC Berkeley, where they worked with Dr. Christopher Schell. Cesar's work investigates how social inequality and injustice, such as historical redlining and socioeconomic disparities, shape the landscapes that both people and wildlife experience. By weaving together ecological theory with environmental justice, the research explores the drivers of environmental equality, wildlife ecology, and biodiversity. In parallel, CESAD works to make ecology a more diverse and welcoming field by creating more mentorship programs, open access resources, and collaborating with other ecologists to integrate the norms within ecology and evolutionary biology. So with that, Cesar, we are so excited to have you. Please go ahead. I'll stop sharing my screen so you can go ahead and get started.
Sweet. Thank you so much for that introduction. Let me pull up the slides. Okay. Everyone can see the slides right now.
Yes,
Thumbs
looks
up.
good.
Okay, perfect. Okay, yes. Thanks for the introduction. As Angie said, I'm at Second Nature Ecology and Design, which is a small urban biodiversity focused consulting firm. And today I'll be talking with you guys about my dissertation research, thinking about how societal inequities, so factors like historical redlining, environmental health burdens, how all that shapes the ecology of cities. So I realize also that some folks may not know what I mean by ecology, so I figured we can begin with a definition and then build our way up. Okay, so what is ecology? At its core, ecology is simply the study of the relationship between organisms and their environment, including other organisms. So things like fox and coyotes are competing for resources, or different components of biodiversity, like how many unique species are in an area, or how abundant each species is, and so on. And so this means that ecology is rooted in understanding how living and non-living things are connected to each other. And so when we're talking about urban ecology, we're taking this definition and thinking directly about how features of the environment, so both societal features and ecological features, shape individuals, including what they eat, how they move, and how they interact with the same species or even other species. And so then the question kind of becomes why cities? Why think about ecology and biodiversity within these urban spaces? So, roughly 55% of the world's population, that's about 4.48 billion people, live in cities today, and that number is expected to increase to 66% by 2050. And as people are moving into cities, that means landscape changes and alterations are happening to accommodate those populations. There we go. That also means that new urban land cover is projected to increase by up to 1.5 million kilometers squared by 2050, which would be like adding new impervious surfaces across the globe that would cover the same area as all the states here shown in gray. And again, that's new gray area, not existing gray area. And so these large numbers, both the impervious surfaces and the concentration of people in these urban areas, are really important to think about in relation to wildlife because over 450 cities are co-located on biodiversity hotspots, which means that urbanization is having a direct effect on the biodiversity within these areas and driving the ecology and evolution of many species across the globe. This also means that the spatial overlap of terrestrial species and humans is expected to increase across the globe by 56.6%. So that means that human wildlife interactions, whether that's positive or negative, are increasing in and around cities as a result of urbanization. And so understanding that relationship between cities and wildlife is essential for building more targeted management and conservation initiatives to maintain and support biodiversity within cities. So early on, questions within urban ecology were really focused on asking how do individuals or wildlife communities sampled in this really urban area differ from individuals sampled in this rural area? So let's think about these general effects that urbanization can have on wildlife and their communities. And so now we know that urbanization which is associated with extensive impervious surfaces, so things like roads, sidewalks, and buildings, habitat fragmentation, pollution, and high human densities can lead to altered wildlife ecology with things like reduction in biodiversity, restricted gene flow, modified behavior and diet, altered species interactions, and more. But as we know, not all cities are the same, right? Oakland, which I live in currently, is very different than San Francisco, even though it's really, really close. And these cities are also different than places like Miami or New York. And even within a city, neighborhoods can vary quite a bit in their ecology. And so moving into this more dynamic framework when approaching cities is really important for thinking about how within city differences and things like income can influence wildlife ecology and how consistent these differences might be across different cities. So, for example, here in the East Bay, where I live, we can look at two different neighborhoods near me that are no more than a 5 to 10 minute drive apart from each other. So we have this neighborhood called Piedmont on the left, and this is a higher income neighborhood and mostly white. And then on the right, we have East Oakland, which is lower income and mostly Black and Hispanic. And kind of immediately just looking at these two side by side, right, we're hit with this really stark contrast of green and gray, right? With the picture on the left of the wealthy neighborhood having what seems like a higher density of trees and more canopy cover than the less quote unquote well off neighborhood on the right. And you can imagine where there are more trees and more canopy cover, not only is it cooler for the people living there when they're going on walks because of the shade, but there are likely more organisms because of more habitat creation and availability. And so this is termed the luxury effect, where species richness and biodiversity are positively associated with wealth. And the luxury effect is something that we see influence and is correlated with many different kinds of wildlife communities, from the bottom of the food chain with vegetation and trees, all the way up to the top of the food chain with birds and mammals, which means that income is dictating where species are, how many species are present. And because biodiversity is really important for things like ecosystem resilience and services, these differences in biodiversity that we're seeing means that people who are living in these neighborhoods are also seeing differences in ecosystem services and resilience for their neighborhood. And even though this pattern is generally true, the opposite pattern can also be found. So, for example, there may be an HOA in a wealthier neighborhood that has strict policies on the type of plant that you can plant, leading to less, flowering plants or monoculture grass, which can then scale up to shape other forms of biodiversity like insects or birds. And so wealth may not actually always be the best predictor of where species are, even though it can be really useful. So this meta-analysis by Curis and colleagues looked at all the luxury effect studies to date back in 2020 and did some partitioning into studies that were in sparse cities and dense cities and looked at residential areas versus non-residential areas. And so they found that when you look at those residential areas, which I have here in that red rectangle, the luxury effect is pretty strong for both trees and wildlife. And so here, light gray means that when there's more wealth, there's more biodiversity. This medium tone gray is when there's no relationship between the two. And this dark gray means more wealth, less biodiversity. And then they found that once you move beyond these residential areas, that the results seem to be a bit more mixed. The luxury effect isn't always super consistent. And when they bend the cities or the studies by the type of city, they found that for plants, that the luxury effect is relatively true, but for animals, the story is a bit more complex, right? It can be disrupted by different things like perhaps city structure, climate, the policies and management within a city, or even human preference, right? People planting what they like and that attracting certain kinds of birds or other forms of wildlife. And this is where thinking more about the role of humans in those social processes within cities can be really, really fruitful, right? As humans, we're deciding what goes where, how connected we want spaces to be, and we make other decisions that ultimately shape the landscape that animals and people are experiencing, and the interactions that people get to have with wildlife as a consequence of those decisions, such that these societal structures, so things like politics, culture, and management, can facilitate or hinder different ecological processes within cities. Much of that can only be shaped by today and the present and what we're doing. So things like economics or culture, but also past decisions by our governments or other neighborhood level decisions that were made yesterday, 5 years ago, 20 years ago, and so on, because all that comes to shape the world that we live in today. And so one thing we can think of, for example, are land use legacies and legacy effects. So legacy effects are these long-term impacts from features that can persist long after their removal from an ecosystem. So this includes structures like the facility I have here on the left and the contamination it can leave behind long after their removal, or agricultural practices on like the one I have pictured on the right, where the type of crop planted, or pesticides used can have lingering effects on soil quality in the community there. And within cities, a huge part of our past and informs the present is racialized segregation. In the United States, one of the ways that was enforced was via the process of redlining, which shaped the concentration and distribution of resources in society and influenced the characteristics of our built environment, which then scales up to shape and affect biodiversity. And so, what is redlining? Redlining was this racialized policy in the United States enacted by local lenders and the Federal Housing Association in the early 1900s, where banks and loaners refused financial services to residents of certain demographics, which predominantly impacted Black communities, but also affected other groups in the United States like Hispanics and immigrant populations. And so in the 1930s, the Great Depression happens, and FDR creates the Homeowner Loan Corporation, or HOLC, as a part of the New Deal. And they're tasked with reducing foreclosures across the US in the face of this economic crisis. And to assist with that, HOLC goes out and they assess a bunch of neighborhoods across the US to evaluate the risk of lending mortgages in certain neighborhoods. And in the process, they create these maps where they grade a neighborhood from A to D. To show you what that looks like, here is a Hulk map from Oakland and Berkeley and in the lesser cities like Piedmont and Albany, where neighborhoods are being graded from the A to D. So grade A, which you can see are in the hills of Oakland, are colored green, and these represent the best neighborhoods. Grade B is colored blue, and these represents neighborhoods that are still desirable. Grade C is colored yellow, and they represent neighborhoods that were definitely declining. And grade D is colored red and represents these hazardous neighborhoods and gave birth to this term redlining. And research has shown that when we look at these maps, which are digitized and are from throughout the 1930s, that the legacy of redlining has these really dramatic consequences for both landscapes and people. So researchers have found that outcomes like disproportionate preterm births, asthma cases, cancer risk, all the way up to more air pollution, less canopy cover, less green space, and more heat in these formulated redline neighborhoods. And more recently, redlining has also been linked to differences in wildlife ecology, predominantly biodiversity. And so here's one of the papers on that list looking at how redlining underpins pollution, air pollution disparities using data from 2010, and shows that on both the national level, which is on the left side, and on the city level, which is on that right column, that minoritized populations and formerly redline neighborhoods are exposed to higher levels of air pollution, looking at both NO2, which is the top row, and particulate matter 2.5 on the bottom row. And another paper showed that redline neighborhoods across the United States, including Los Angeles, Seattle, and Philadelphia, also have less canopy cover, which again can lead to warmer neighborhoods. And on the wildlife side, work published out of Los Angeles showed that redline neighborhoods have lower amounts of forest-dependent birds, which are more charismatic bird species. So thinking of the warblers, jays, woodpeckers, and higher levels of synanthropic birds, so birds that are more dependent on human subsidies. So species like pigeons, starlings, and house sparrows. And so we can begin to understand that urbanization as this process and mechanism is inherently driven by different logics of oppression, such that structural racism and classism via segregation and differences in resource allocation can contribute to how uneven cities are with respect to things like pollution, heat, green space, impervious surfaces, which all have potential to shape different ecological processes like biodiversity and food webs within cities. And so my dissertation research kind of starts here and centers inequity as this origin of urban heterogeneity or differences in the landscape to explore how inequity shapes the ecology of cities. And so we'll start with my first question, which really wanted to get an understanding of how is redlining shaping the landscape across California and how does that relate to the urban ecosystem health of cities in California? And this was done over two different parts. So first, looking at differences in environmental quality, and the second, looking at patterns of wildlife biodiversity. And so first, we'll be looking at if redlining shapes environmental quality to kind of ground truth our hypotheses for why biodiversity should even vary along these different Hulk grades from A to D. And so in California, 8 different cities have their redlining maps digitized. If you're curious, the Mapping Inequality Project has digitized it for over like 150 cities from the University of Richmond. So you can go and look at the redlining map for the city that you live in. And so in California, that includes Los Angeles, San Francisco, San Diego, and Oakland. And then I use this environmental hazard screening tool called CalEnviroscreen, which maps different environmental hazards and effects down to the census tract level throughout California, along with different satellite data sources and a data source called How Loud to explore this spatial association of environmental hazards with these redlining maps. Then I asked two questions. So the first being, is redlining associated with environmental quality across California? So asking, do redline neighborhoods have more of these environmental hazards relative to these other grades? But then also asking, do redline neighborhoods face higher negative intra-urban disparities in environmental hazards? So asking in relation to their respective city, are these redline neighborhoods worse off? Are they experiencing disproportionately more of an environmental hazard? I did this across 10 different metrics, including lead risk, particulate matter 2.5, and noise pollution. And then I followed CalEnviroscreen methods and I created this larger pollution burden metric, which sums all these metrics shown here in black. So everything besides vegetation, temperature, and noise levels, and then bend that into percentiles, which is what I'll show you going forward. So closer to 0 means that you're on the lowest part of experiencing a environmental hazard, and 100 is the max amount. And this work was published in Environmental Science and Technology Letters if you would want to read more about it. Okay, so first, after controlling for area of a neighborhood, right? So bigger neighborhoods might just have more pollution simply because they're bigger and they encompass more of an area. Non-green line neighborhoods have more environmental hazards on average, as denoted by those diamonds, with Hulk grade on the x-axis and the pollution or hazard of interest on the y-axis. And this remained consistent across almost every pollutant that was put into that pollution burden category. So air pollution, groundwater threat, lead risk, and so on. We also see this pattern is true for noise levels, with redline neighborhoods having more noise pollution than the other grades, having the least amount of vegetation, and also having elevated temperatures compared again to those green line neighborhoods. Now, this is overall environmental hazards present in a neighborhood, but again, it's important to think about those city level differences because, for example, the San Joaquin Valley in California, which has cities like Fresno, has some of the worst air pollution in the country compared to a city like San Francisco or San Diego, which doesn't experience nearly as much air pollution. And so a red line neighborhood with an average percent of like 80% air pollution burden can mean something really, really different in a place like Fresno compared to a San Francisco. And so I calculated the city average for each pollutant and then plotted the difference between a neighborhood and that average to think about within city, these intra-urban disparities. And we found that across each of these metrics, that these red line neighborhoods face disproportionately worse hazards compared to green line neighborhoods across California. So 76% of red line neighborhoods have more pollution, 72% of these red line neighborhoods have higher noise levels, 86% have less vegetation, and 72% of red line neighborhoods are warmer than their respective cities, compared to those much lower proportions that we see in these green line neighborhoods. And if you want to look at this spatially, here's a map of Los Angeles with different environmental hazards. We can begin to see how these red line neighborhoods have worse environmental quality concentrated in their areas, and how they may also experience diminished wildlife biodiversity as a consequence of these concentrated environmental hazards, like more noise, more pollution, less vegetation, and so on. And so next I want to kind of build on those results and to see how the patterns may change with Holt grades because of these concentrated environmental hazards, right? And so I predict that these red line neighborhoods have a lot less wildlife biodiversity than these grade A neighborhoods or even grade B. And so to do this, I worked in 4 cities in California based on data quality from iNaturalist, and I worked across 6 different terrestrial taxonomic clades. So mammals, birds, amphibians, reptiles, insects, and arachnids. And we asked 3 questions. So first, I wanted to use the bias built within iNaturalist, right? So iNaturalist is an app that you can download on your phone, and it's not structured, so you can go anywhere, take a picture of an organism, and report it to the cloud. All right, so there's different biases, which I'll talk about that in a few slides. I want to control for that kind of user and observer behavior and think about species detections, which could be due to differences in participation across these grades, but also better environmental quality could also promote the usage of iNaturalist because you're encountering more wildlife in your neighborhood. And it's nicer to take a picture of that bee on that flower. And then after controlling for the biases in the data and the negative effect of urbanization, we want to know if these redline neighborhoods have lower species richness, and if that, it was consistent across each clade. So are there less mammals and less birds and less insects, or just less birds, but no effect on mammals, for example? And then last, we want to know if these redline neighborhoods had an altered species assemblage or differences in the types of species present within a neighborhood. And if you want to read more about this work, this one was published open access in the Proceedings of the National Academy of Sciences. Okay, so focusing on that first question of differences in species detection, we found that individuals, and I'll walk through this graph, there's a lot to digest. Well, we found that individuals in these green line neighborhoods detect species with less sampling effort in these California cities. And so on this graph, we see that these green line neighborhoods don't have the most observations, right? So they don't go super, super high on the x-axis. And we can also see that these green line neighborhoods don't have the highest amount of total observed richness or the most observations. So on the x-axis, they're not going super far out on the right for observations and not super high up on the y-axis for richness. But when we look at how fast it took grade A to reach its max richness, which is denoted by that horizontal dashed line, grade A neighborhoods reached this in about 17,000 observations, which is shown by that first vertical line, while it took almost an additional 8,000 observations to detect the same amount of species in these red line neighborhoods, which is shown by that second vertical line. And this suggests that individuals in green line neighborhoods are detecting unique species with less sampling effort than individuals in other Hulk grades, not just red line neighborhoods, which could be because of higher vegetation and more tree canopy in these neighborhoods. And so I like to think about this in the way that if you were to go on a 10-minute walk in a green line neighborhood with more vegetation and canopy cover, the likelihood of me running into a different mix of pollinators, so bees, butterflies, hummingbirds, other kinds of maybe more charismatic bird species that like dense shrubs or mixed habitat types, that would be higher than in these redline neighborhoods that have less vegetation, more impervious surfaces. Okay, so next we wanted to control for these differences in observations that we see here on this graph, the size of a neighborhood, and also that negative effect of urbanization to see how species richness varied among these grades. We found that across each city except for San Francisco, that these redline neighborhoods have the least amount of species richness, which remained true even when we disaggregated our data into native and non-native species. And interestingly, we found that these green line neighborhoods did not always have the most species, as shown in Los Angeles and Oakland, on the top row here. But still, we're kind of seeing these consistent differences between green and red line neighborhoods, with green line neighborhoods having significantly more species than these red line neighborhoods. And we also see these much larger differences in San Diego and San Francisco compared to Oakland and LA, which we think may be attributed to greater differences in habitat complexity, for example, between grade A and D. And so not just vegetation or the presence of trees, but other factors. So thinking about understory, vertical habitat diversity, and so on. And so next we wanted to see how true was this pattern for each clade in cities because of differences in life history and movement across these taxa. So for example, amphibians or insects, right, they have really small home ranges compared to a bird. And so you might not expect to see differences in bird richness perhaps between these grades because of movement, but you may see differences for clades that have smaller home ranges. And so when we disaggregated this by clade, we found that in Los Angeles, in Oakland, San Diego, and in San Francisco, that for each clade, the red line neighborhoods generally have the lowest amount of species richness, with variation again on which grade had the highest richness, but still we were able to observe those consistent differences in biodiversity between green and red line neighborhoods, with these differences remaining again after we disaggregated the data into native and non-native species. And we also see these trends here for arachnids, reptiles, and amphibians, which I don't have shown here. Okay, so next we measured beta diversity, which looks at differences in species assemblages. And we found that these green and red line neighborhoods have distinct species assemblages in each city, and this again remains after you disaggregate into native and non-native species. And so in these graphs here, each dot represents a neighborhood, and each circle is a 95% confidence interval. And what really matters for this graph is thinking about the overlap in the size of the circle. That tells us how similar or distinct the wildlife community is relative to other circles. So larger circles indicate a more diverse species pool. And if the more two circles overlap, that means those species pools are more similar. So we focus on just Los Angeles and San Francisco here. We can see that these red line neighborhoods are have a smaller wildlife community than these green line neighborhoods, right? So it means it's not as diverse. And because they sit within these green circles, it suggests that the species are similar and that these red line neighborhoods likely hold a subset of urban species that are more maybe tolerant of environmental hazards I described earlier. So wildlife like pigeons, house sparrows, and raccoons. And while, a Greenland neighborhood may also have those species, they may have additional ones like foxes, California scrub jays, and so on. Okay, so now that we know that there are differences in biodiversity across neighborhoods within a city as this function of redlining, folks may want to start thinking about how do we bolster biodiversity and target these redline neighborhoods for restoration initiatives. And the first step to do that would be understanding what species are even present in these neighborhoods. And so theoretically, this shouldn't be too hard to do because we live in a digital age where apps like iNaturalist and eBird go beyond professional collection to get us additional fine-scale data that stretches globally on the biodiversity we have, including birds, plants, and insects. And so here I'm showing you all the data found in the Global Biodiversity Information Facility, or GBIF, which holds biodiversity records for collections and human observations on apps like iNaturalist, with more data being reflected by that deep red color. And yet, still, we're seeing that societal inequities and different legacies of injustice are diluting our ability to understand different patterns of biodiversity. So, of the over 2.6 billion species observations in GBIF, we see that these observations are disproportionately from higher income countries, and that these disparities in data density have become more pronounced throughout time. We also see that redline neighborhoods have less than half the amount of bird observation density compared to the neighborhoods that were deemed a quote unquote safer investment, so grades A through C. And when I looked at this for three cities on a much finer scale, so here I'm showing you data just for Baltimore, but we also looked at Oakland and St. Louis. We found that generally that these socially privileged areas, so census tracts with more income and more white people, not only have more observations, but also more than you would expect based on population density and area. And so these areas are almost in like a data surplus compared to data deficit in lower income areas. And so we can look at this spatially in these examples in Baltimore and St. Louis. And we can see that inequity or differences in wealth and differences in race are spatially associated with biodiversity data, and also the gaps between what your report and what you might expect to see there. And if we look at these maps further, we can kind of see that there are even more biases present within these data. So if we just look at St. Louis on the right, we can see that most of the data, again, is concentrated on the southern side of the city where there's more income and there's more white people. So in St. Louis, there's something called the Del Mar Divide. You can kind of see that very clearly dividing the city in the middle and also the data. But if you look closely, you can see further clumping in this data. And so there's additional social or ecological factors that may be leading to further biases in the data. And so to think about how that clumping happens, we have to zoom way out and think about how an observation is reported. And so to start, you're in a city within a given biome, right? That determines your actual species pool within a given habitat. So here in Oakland, for example, simply put, I would not expect to walk outside and see a tiger or a polar bear, right? That's not the biome I'm in. That's not dictating the species. That is dictating the species pool that I would see when I step outside. And so in this city here, we have a mushroom, an owl, a snail, a coyote, and a few other species. But as we know, there are differences in participation that I just showed you guys. And so we saw that higher income neighborhoods have more observations than lower income neighborhoods, or that red line neighborhoods have less than green line neighborhoods. And so we'll say that anything that's not in this red circle right here is not going to be reported to iNaturalist. So for example, again, we know that because people are participating differently across neighborhoods. And so we'll lose that snail that might be in a lower income neighborhood. And we're also going to lose that reed and that fish along the stream. Okay, so now we're within that biased sampling area in a higher income area, or maybe in a green line neighborhood, and further biases continue to reduce this actual species pool to what we download when we're looking at biodiversity data. So first, we kind of describe this detectability filter, which we highlight that people can only report what they can see, which may be a result of a species biology or behavior which impacts their detectability. So nocturnal species, timid species, cryptic species that are harder to observe. So in this case, the owl in the tree, the salamander, and microorganisms in a water feature are not going to be reported to these kind of platforms. Next, we talk about the sampling filter. And here we emphasize that people participating are generally more willing to sample in green spaces while walking or recreating than in these gray spaces while commuting, like downtown, for example. And so this means that species that are more common in gray spaces, so street trees, pigeons, and raccoons, are just less likely to be reported or have a lot, lower observations total compared to another species that may be more charismatic or found in green spaces. And so if we look back at St. Louis, we can actually see this sampling filter at work with observations clumping on eBird and iNaturalist in places like Forest Park, Gateway Arch National Park, and other parks throughout the city, even though people in canopy cover are pretty well distributed throughout the city. And lastly, we encountered what we're calling a preference filter. And this reduces the pool to a charismatic, rare, flowering, and colorful species. And this excludes these nuisance or boring species, quote unquote, like rats, woody shrubs, non-colorful plants, non-colorful mushrooms. And this filter captures the fact that people are more excited or drawn to more charismatic wildlife and flowering plants and are more likely to take pictures of them and report them to these platforms like iNaturalist or eButterfly, for example. And then that all brings us to the data that we're going to get to download, where all the blacked out species are what we've lost at each filter, which does not include the species that are not found within that spatially biased sampling location. And so here we're left with 5 unique species, and that includes the coyote and the butterfly and that flower right by the pond. And so we're seeing that data is ultimately shaped by social biases, such as who is participating, where do they select for, and what do they select for, ultimately leading to these spatial gaps in data and shaping what neighborhoods have more biodiversity data, which shapes what kind of work for conservation or other ecological work can be done in that neighborhood. Okay, so take a little step back and just recap what we've seen so far. We've seen that legacies of injustice and inequity can shape the landscape that wildlife and people experience with more pollution and poor environmental quality in those red line neighborhoods. It can influence urban biodiversity with less overall wildlife biodiversity across different taxa in those red line neighborhoods. It also creates these gaps in the biodiversity data. And so to continue that thread for the last part of my dissertation, I was interested in exploring how urban heterogeneity and environmental quality and urbanization can also have consequences on organismal biology. So thinking about different environmental pollutants and the built environment itself via impervious surface cover. So for instance, animals exposed to environmental contamination can display more risk-taking behavior. It can have these consequences on their behavior. So fish, for example, when individuals were exposed to pollution, spent less time being vigilant due to a degraded ability to recognize predators and novel objects in water. Additionally, research has shown that individuals exposed to contaminants have poor cognition and problem-solving abilities. So honeybees that were exposed to metal had poor memory and learning, and in rats, those exposed to air pollutants were slower at completing mazes. And lastly, animals exposed to contamination also show differences in movement patterns and activity levels. So birds that were exposed to a PCB mixture showed differences in diel activity and also orientation for migration. And so since pollution is unevenly distributed in cities, it's plausible to think that these species in more polluted areas may show behavioral differences as a consequence of living in a more polluted neighborhood. And as shown here a little bit, most of our knowledge for how contamination affects individual behavior lies in a lab-based controlled setting with little field examples, and is also skewed towards a lot smaller organisms. So little is known about how the behavior of larger animals like coyotes or raccoons are impacted by pollution. And so to explore this question, I set up some cameras in the East Bay. And at each site, we put down an attractant, which was composed of a fatty acid scent tablet and some meat, which was our control. And then after 4-ish weeks, I came back, I rebaited the site, and then I set up a novel object, which you can see here is four wooden posts with a string wrapped around the top, kind of creating this open box which animals can move through freely. And this was similar to a study done by Breck and colleagues looking at coyote boldness in Denver. And so novel objects are this common tool in behavioral ecology to measure boldness and exploration because novel objects can transform a familiar environment into a riskier one by introducing a neutral but unfamiliar stimulus to a familiar environment. So here I'll show you a couple of videos. So here on the left, we're seeing coyotes sniffing our bait at the control, right? Scent marking, which is very normal behavior. They're spending time close to that attractant in the middle. Um, they're exploring it, they're sniffing it. They don't seem particularly cautious about their environment, for example, right? The second one is also scent marking, which we would maybe classify as like a comfortable behavior. And then 4 weeks pass. I go back, I set up the novel object, and we can kind of see before I even hit play on the video that this coyote, which is likely the same coyote, is now crouched, right? Its ears are tucked back, its tail is a little bit tucked. And so we're seeing this immediate change in behavior from this coyote. So this video will be a little bit shorter with the coyote on the screen, but you can see very quickly moving through the frame. It was crouched, kind of keeping its distance, not getting close to that attractant anymore, and staying crouched. And so this behavioral change is something that we would generally expect to see in coyotes, actually. They are pretty cautious of new things in their environment, but within cities specifically, coyotes have been shown to be bolder and more exploratory than those non-urban counterparts. So the video I just showed you was taken at a less developed site, for example. So this study from Breck et al. Showed that in urban areas, which are these gray boxes here, that urban coyotes have a lower flight initiation distance, meaning that you can get closer to them before they run away from you, compared to the rural counterparts shown in that white box. They also found that coyotes in their study area spent more time investigating and sniffing that novel stimulus, so both during the control and the object, even in the presence of a novel object, compared to their rural counterparts. And as for raccoons, that relationship between urbanization and their risk taking is relatively unknown. Right? There aren't really any studies that compare non-urban and urban raccoons or raccoon behavior along an urbanization gradient. But when we observe raccoons and coyotes in a novel foraging task that's similar, we can kind of infer that raccoons are bold and exploratory species and that they're riskier than coyotes. So in this example study here, raccoons interacted pretty much immediately as soon as presented with the novel foraging task, whereas coyotes did not interact until like the 40th trial. And so at each site, we deploy that novel object and we're coded for a set of behaviors that reflect risk taking. So we focused on time spent alert. We focused on time within one body length of the object, the amount of time spent sniffing, the total number of exploratory behaviors an animal displayed, which measure the amount of times a coyote or a raccoon touched the object, it dug, it sniffed, and so on. And this work want to highlight would not have been possible without an amazing team of undergrads that watched a lot of videos of coyotes and raccoons. So for each site, I made a buffer around each site of about 500 meters. I did about 250 for raccoons because they're smaller with smaller home ranges. Then I extracted the average pollution burden from CalEnviroscreen, which was a data source I used from that first chapter I showed you guys, and is composed of 13 different metrics, including air pollution, lead risk from housing, and contaminated water bodies. And then I also included human population density and activity as another variable of interest, as there's a lot of studies in mammals, birds, and so on that have found relationships between human population density or activity and elevated boldness in exploration and wildlife. And then each site was categorized as having high pollution or low pollution, and high human density or low human density based on the median in our data set. And so for this project, I was interested in how urban heterogeneity influenced coyote and raccoon ecology, focusing on the amount of detections for each species and those risk-taking behaviors. And since coyotes can be in pairs and raccoons are often seen in large groups, I was also interested in group size and how that influenced their behavior. So for coyotes, I'll be showing you guys how does human density, pollution, and group size influence their risk-taking and detections. And for raccoons, but doing the same thing, but also adding how group size influence their behavior in relation to urban heterogeneity as well. So first, we'll be looking at the detection results for both species and then the group size results for raccoons, and then we'll go into behavior briefly. Oh, and there's that other point. Okay, so for coyote detections, we found that in areas with more people and more pollution, that there were less coyotes detected relative to the areas with less people and less pollution, which can suggest that there's a mix of factors associated with people and pollution, so like limited green space, less vegetation, more opportunity for human-wildlife conflict and the subsequent removal. All those things can limit their establishment in these really dense areas and also really polluted areas. In contrast, we saw that raccoon detections were high in areas with lots of people, both density-wise and for activity, which we measure by how many people were crossing our cameras. And this makes sense given the amount of anthropogenic subsidies and resources that raccoons can access by being in areas with more people and more human activity. And although we found that raccoon detection, so the amount of times a raccoon walked in front of the camera, which can be a proxy for activity at a site, was correlated with more people, we actually found that the size of a raccoon group decreased at sites with more pollution, which suggests that these really polluted areas may limit group size and group aggregations due to different pathways, such as resource limitation, a low predictability of resources. So when people are dumping trash, that doesn't happen super frequently or lower predation risk, right? There's less things that might have agonistic interactions with them and reduce the need to be in a larger group. Okay, so now we'll talk about coyote behavior. So we found that human densities and group size is what influence their behavior rather than pollution. So here I'm just going to highlight the human density result. So we looked at the control, so that's the attracted only and captures a baseline behavior, and that's these black lines shown on these panels here. We found that coyotes at these sites spent more time alert, more time closer to the attractant, and also explored our attractant more, which we think is pointing to the fact that coyotes in these higher human density areas may be less interested or inquisitive about the attractant, likely as a result of habituation to these novel scents. And we also see that coyotes explore more when they are in a pair. And when we compared the treatments to each other in different environmental contexts, we found that only the coyotes in these low human density areas were adjusting their behaviors in response to the novel object by reducing their vigilance, spending less time close to the attractant, and exploring less, pointing to, again, that increased wariness and caution in these lower human density areas I showed you guys in the video, compared to high human density areas where they seem to be not fazed by that novel object or change in their environment. Now for raccoons, the story is a bit different. So here I'm only going to show you the figures that highlight the results for exploration, since we have a lot of different figures for raccoons. So first we saw that raccoons in larger groups explored more and they spent less time alert during the control period. And that sites with more human activity, raccoons increased their exploration, whereas sites with more impervious surfaces, they actually explored less. And we saw pretty much identical results for the object period, except we saw no differences between sites with human activity. And so overall, we're seeing that raccoon exploration is being bolstered by group size and human activity and dampened by sites that are really developed with more impervious surfaces, um, which can be explained by differences in, perhaps mechanisms like habituation to novel objects or sense, or individuals that explore more in these more developed areas from the population might be removed by things like animal control, leading to less risky phenotypes in that area. Whereas at sites with more human activity, there might be different human subsidies, trash left behind, all things that promote more exploration. And so for the coyote and raccoon work overall, we saw that coyotes are less detected in areas with more people and pollution, whereas raccoons are found more in areas with more people and more human activity. But their group size gets smaller at sites with more pollution. So let me play these cute videos of a coyote rolling around and some raccoons. And we found that for behavior, that human density and group size is associated with riskier coyote behavior, whereas in raccoons, human activity and group size increased exploration, while impervious surfaces reduced the amount of exploration a raccoon did at a site. Do, 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 do. I want to advance. Okay, there we go. So across all these projects, kind of highlighting the ecology in cities and how it varies based on social and ecological factors, I hope I got to highlight that there's this really high interconnectedness between social inequality, such that it is an ecological issue, and that injustices shape our landscapes, both natural and social. With that, I'll wrap up another cute video of raccoons, and can take questions.
Awesome. Thank you so much, Cesar. I can hear it from my office. There's a lot of applause for you in our lounge right now. Um, so yeah, we'd like to open it up to the audience for questions. You can leave your questions in the Q&A button at the bottom of your Zoom screen. And we'll wait a little bit to get some, but in the meantime, I will ask a question. Um, so kind of going back to the first half of your talk, cities are obviously like very highly dynamic over time with construction and redevelopment and green space turnover. So I'm curious if you've looked at how well our current biodiversity data sets capture this temporal change, and if you think that we're missing anything by relying on mostly static snapshots.
Yeah, that's a good question. Um, I think INAT data, eBird data, GBIF does a pretty like birders are just an example I'll highlight. Birders love to bird. So like bird data is so current that it reflects, yeah, all these really nice patterns. A colleague of mine just, put out a paper showing how you can use eBird and these data sets to even track things like mortality because of how often people are using these data sources to take pictures of wildlife. So um, yeah, I think that our GBIF data, it can be a little static depending on like certain species. And so it's not always the cleanest answer. And that's where having to work with local groups is really important often, and local experts to understand certain species because things like a coyote maybe, or I don't know, like a cardinal, California scrub jay, is super, super overrepresented in these data, but like a really specific species, you might not be able to get a lot of information for. So that might be static just because it's super rare.
Yeah, thanks so much for that answer.
Mhm.
Still not seeing anything in the Q&A, so I can ask another question unless somebody else has one. Oh, question from Erica. All right. So, with respect to historic redlining and its effects on biodiversity and biodiversity monitoring, what's one result you might not have had time to share but would want a layperson to know?
Sorry, can you repeat the question? I was reading a different question.
Yeah, you're good. So with respect to historic redlining and its effects on biodiversity and biodiversity monitoring, what's one result you might not have had time to share, but you would want a layperson to know?
Wow, that's a really good question. Um, hmm. Yeah, I'm trying to think of what I left out. I have a habit of like cramming everything into a paper or having separate papers. So I think one thing that started to come up while I was doing this paper were the things around biases. So like that's why I had like two different papers on that. Um, yeah, so I think what I've communicated to, like non-scientists who like love INAT is like everything is valuable when it comes to taking pictures, even things you might not think are cool to take pictures of, like a possum or even things like dead animals on the side of the streets are really useful just for understanding again these patterns that can show up because often like in my job, when we are going to a new site and we're being asked to think about wildlife, the first thing we will always do is look at the INAT data or the GBIF data at the site and around the site. So every piece of information matters. It doesn't really answer the question so much just because I feel like I actually I crammed everything into that paper and other papers. Um, but yeah,
No, that's a great answer. Most of the questions I had for you were about biases, and you answered them so well that I wasn't going to ask any. So
cool.
great job. We have two questions in the Q&A. Do you want to pick which one you want to answer if you can see them?
Yeah, I can answer the first one. Are there particular strategies that would be most effective for improving urban biodiversity? Yeah, I think different kinds. I mean, so the clearest answer usually, or most questions I get are like So we should green these areas like immediately, and then also like people should go and collect data for these neighborhoods essentially. So I think my response usually to that is like flipping it and thinking about how do you empower these groups to, or people who live in these neighborhoods to want to sample more, whether it's by like curating a neighborhood coalition that is more interested in getting biodiversity data, and thinking about like how do they want to green their neighborhood, right? Greening from like a city perspective sometimes can lead to like different forms of gentrification. So how do you make sure like these neighborhoods are empowered with toolkits for greening or different management to make sure their urban biodiversity is improved while their environmental conditions are improved so that they don't, get displaced from their neighborhood. But I think most effective, it's always like it is like bottom up often. So like even simple changes by like swapping out non-natives that people like with native species, planting annuals, really easy things that people can do, like spreading annual seeds, are all effective for like attracting more pollinators, for example, which might bring in other species as well. So I think that was that one. So quick answer
Yeah,
live.
awesome. And we have one more in the Q&A, which says, my understanding is that redlined areas also have disproportionately high highway or freeways nearby, and that these large roadways act as large bottlenecking. Have you found these two things come out or are related to each other in your work?
Yeah, in my work, I didn't do a ton of like genetic kind of testing, but some colleagues of mine did some work in Seattle and Tacoma with coyotes and showed that like the highways there, they kind of formed this like cross that the coyotes are kind of more genetically like there is like a severance of just connection and connectivity across these areas in terms of gene flow. So that's like a really cool paper that exists. And here in Oakland, there's another cool paper looking at highways and air pollution and showing how like different roadways and truck movement laws around diesel trucks, for example, are influencing air pollution in certain areas and not the others. I think one reason why I am seeing less coyotes in the flats, are just things about park size, but also thinking about highways and structures that are perhaps limiting dispersal from the flats where they mostly are in those Greenland neighborhoods down into the more or the hills down into the flats. I would say they're definitely related.
Awesome. So we are almost at the hour. So we want to wrap up by thanking you again for a fascinating talk and conversation. Thank you so much for presenting. And to everyone online, thank you for joining us in our second talk of the NC's seminar series. I'm going to steal the screen really quick. So for those of you who want to stay in touch with NCs, you can reach us at various places online, like our website, our Instagram, and our LinkedIn. The best way to stay connected with us is through our monthly newsletter. So there's a QR code you can scan here to subscribe, and contains updates on things that are happening around NCs, funding and proposal opportunities, new papers, and much more. And then coming up, we have our last talk of this year's seminar series on June 11th, where Patricia Balvanera will discuss plural syntheses of knowledge to support more just and sustainable futures. And we also have a webinar on June 23rd for our new Luna initiative, Call for Proposals, which aims to turn promising ideas into actionable directions for science and decision making by bringing groups together to work on high risk, high reward ideas. So thank you all so much for joining. This recording will be up on our YouTube channel, and we will share it with the attendees afterwards. And thanks, Cesar.
Thank you.